putting our nuclear notes, finishing our note packet up. Remember, you're going to turn that in next time along with your daily work. So we'll correct daily work next time. You'll turn this in next time so I can record it. If you still want it to study, I'll have it recorded by the end of the day next time, and you can use it to study for your SAGE test. Okay, so half-life. Half-life is the amount of time it takes for half of it to be left. That's what a half-life is. Okay, so half of the sample is going to decompose. The rate of nuclear transformation depends only on the reactant concentration. It doesn't really care about the final concentration. Oh, went too fast, sorry. So I start with 20. Half of 20 is 10. Half of 10 is 5. Half of 5 is 2.5. And I just keep going down. So it's a nuclear decay. Anybody recognize this plot from math? What's this name of this kind of graph called? It's called what? Okay, it's a bar graph. I'm more thinking about what does the shape of this graph look like? It's exponential decay is what they call it. Okay, so it's exponential decay. So we test for radiation using a Geiger counter. That's probably, if you've ever watched Ghostbusters, that's what they're sort of looking at for the ghosts. Okay, that's actually a Geiger counter. They've got a Cillian counter, and you'll also have seen, if you've gone to the hospital to get any x-rays done, that they'll be wearing a film badge. So there's different ways we can test for exposure to radiation. So if I have a sample of radium-234, and its half-life is 3.6 days, if I start with 50 grams, okay, if I start with 50 grams, after 3.6 days, I get 25. So it takes 3.6 days to get half of it left. After another 3.6 days, 7.2 days, I have half of that left. After another 10.8 days, another 3.6, I get 6.5. So you can see how this works every time I cut in half. Okay? So let's look at this one. Let's think this one through. If I have a half-life of 13 hours for iodine-123, how much of a 64 milligram sample of iodine-233 is left after 39 hours? So that's probably the first thing I need to do. How many times will 13 go into 39, right? I need to know how many times I'm going to cut it in half. And this one is just perfect. It will go in 3 times. Now, it won't always be so nice. Okay, but this is 3 times. So I'm going to cut 64 in half 3 times. So 64 in half once is what? 32. In half again, 16. In half again, 8. So I have 8 grams left after 39 hours. Okay, when we talk about radiation, we also talk about how does it affect our body. And this is really important. If I have 0 to 25 rem, there's no effect that can be observed. But if I go to 26 to 50, I get a small decrease in my white blood cell. If I go to 51 to 100, I get a real significant decrease in my white blood cell count, and I get lesions. Anybody know what lesions are? They're basically sores. I get sores that start to appear. If I get to 100 to 200, I get loss of hair and nausea. Wait a minute, what does this sound like? Chemo. It sort of sounds like chemo. This is what radiation they do to kill parts of their cell. But if I go past that at 200 to 500, I get hemorrhaging. Anybody know what hemorrhaging is? Bleeding out anywhere. I can bleed out anywhere. I get ulcers and death in 50% of the population. And if I'm above 500, I get death. So when we talked about Chernobyl last time, they had an exposure above 500 rem after they had exposed, after they'd flown over. And so that's why they died. Okay? So how does a Geiger counter work? Basically, you've got some pictures. I think they might be on the second page. I might not have gone in order here. They're down at the bottom. So you've got different things that you can do. So I've got a Cillian counter here that basically brings in, and it has a thing out here that grabs counting. If any of you have anything from Mexico that has red finish on it, those will most likely be radioactive. We can actually take a Geiger counter and test it. The Salt Lake Temple is made of granite. Granite is radioactive. So you can go up to Salt Lake Temple and hold a Geiger counter, and it will go off. Okay, yes. Best way, G, I always get the E and the I wrong. Yeah, so it's right where? Sorry. There we go. See, I had to look it up. Okay, so radioactive things are all around us every day. And so to say that, you know, we have problems with radioactivity, we're breathing it in every day. So cosmic radiation, just because we get 50 millirem a year, just because we're alive and we're living in the cosmos. On the Earth, just because we're on the Earth, we get another 47. If we're around any kind of building materials, if we inhaled the air. So you can see there's some things that it just happens. We actually eat salt. Salt has radioisotopes of iodine in it. So we're eating stuff that is radioactive. So we get this much every year, just because. Now, if we end up getting an X-ray, you can see that they're really careful about how many X-rays that you get a year. Why? Too much radiation. And they're really careful about how close to each of them you're getting it, because after time that'll get out of your system. Artificial sources. So if you look right here, down at the bottom here, everybody's so worried about nuclear power plants. Wait a minute. Nothing else is point something, right? It's just because it's so contained. If you watch TV, fallout from nuclear tests, for us that's really high, because in the West Desert they did a lot of nuclear testing and the downwinders and stuff like that. So there's a lot of problems here. You can see clusters of brain cancer. There's actually a cluster of brain cancer that is up by the burn plant that they've been watching. And is it because of the burn plant, or is it because of stuff that happened at Hill before and that wasn't developed? So they don't know why. Okay, so radiocarbon dating. This is how we can find out how old something is, and it's being really slow. So this is a neutron experiment again, and I probably should have put gamma on the end. Radioactive carbon is formed in the upper atmosphere by reactions. Okay, the C14 is oxidized into CO2, which circulates through the biosphere. Plants take up that CO2 while they're living. When they die, and when anything dies, it stops taking up this C14. And so the C14 starts to decay. And because the C14 starts to decay, we're able to see how long something's been dead. So we compare it to something that's found nearby that is alive, and we test the C14 in it and the C14 in something that's dead and compare them. The half-life of C14 is 5,730 years. So it's good for dating things that are very, very old. However, it's not so good for dating things that are a little old, okay, because there's a lot of variation. If you looked at that curve, it went down really fast at first and then sort of flattened out, right? Okay. We call this activity of the sample. Okay, when you go into imaging, we talked about this a little bit last time. If you have a healthy thyroid gland and they use technetium-99 to see what your thyroid looks like, it's like a butterfly that's right here in your neck. Okay, if it's healthy, it looks like this. If it's not healthy, it has some different effects when they're looking at it through the X-rays. Mushrooms, we talked about food being irradiated to make it farther and last longer. If something's not irradiated, then the bacteria and stuff on the mushroom itself will start to decay. Once they're irradiated, it kills those bacteria, and you just have the food that's left. So for irradiation, we use cobalt or cesium. And just a few years ago, they started irradiating milk so that it could be just stored on the shelf. I think there's a Gossamer's, and they just come in the little boxes, and you can find them on the shelf. You can chill them because they taste better chilled than warm milk, but they can last for a longer time without radiation. And they've approved it for meats and eggs also, so they can last longer. So when we get to artificial elements, ooh, cool, I can make gold. Look, I can make gold. Isn't that awesome? It costs a lot to make gold that way than it does just to find it and refine it normally. I mean, a lot, a lot. And a few grams of gold that it will cost me a room full of gold to make it. Okay? Okay? All right, this right here is called the Fermilab. And you can see we talked about accelerating. There's two circles, and they accelerate the particles up in the two circles, and then they open up a pathway between them and smash them, and they're able to make new things. Yes? Mm-hmm. Yep. Sorry. Sometimes I just buzz past things. And we'll go do some of the first part. I noticed that I hadn't put it in. Do we get that all? Let me know when you've got it. We'll move on. Good to go? Yes, sir. On which one? Top? Probably because it needs an extra something. We got, I don't know. I don't think it does because 239 and 12, that's 251, right? If I'm adding right. And that should be californium. And let's see, uranium is 92 and 6. 92 and 6 is 98, right? So we're good. Californium. This one shouldn't have the N in it, so just erase it or scribble it out. That should be a U. Yeah. Okay. So we talked about the fermium lab. And this is inside the fermium lab, and they have to keep it fairly cold, closest to absolute zero as possible, okay, to try and control when they hit each other. This is at Stanford. This is their linear accelerator, so they just accelerate them at each other and smash them together, sort of like two cars, okay. Weird guy. And I think I brought up the other one. The one that, I brought up the old one instead of the new one. That's okay. That's why it didn't follow your notes. Hello, I'm finally getting it. All right, so let's go back and let's bring up your notes with, oh, my brain. Okay, with your assignment. So at the very first of your notes, I put this on your assignment, so it's there. This right here, okay, these equations right here at the very top are going to be very important to you, and I've actually defined them. So if you want to rewrite them in your notes, that would be good, or just put C, daily work, they're there. T is total time, T1/2 is the half-life, and N is the number of half-lives. So when we found that 39 hours and the half-life was 13, we just basically plugged it into here to find N. So N would equal total time divided by T1/2, right? So this is on your daily work, assignment 3. Okay. This right here, AI and AF, I can't see the difference between the two really easily here. AI is on this red right here. This is initial. This is the amount I originally have. So it's either amount or it's count. Sometimes we use counts instead of the amount because we don't know the amount on some of them. We'll take a Geiger counter count, and then a few hours later we'll take another Geiger counter count, and then we can figure out the half-life. So if I'm trying to find N, and all I've been given is the initial amount and the final amount, then I have to plug it into this equation where I do the log. Okay? So I rearranged it for you, and you just have to plug in. And it's the ln, it's the natural log. So let's look at this first problem here. And I know I didn't give you a whole heck of a lot of room there. So I'm going to go to the next page here and put it up and give me a little bit of room. So I'm going to put it down at the bottom. Okay, so I'm going to pull this up so we have some room to write. Okay, so I begin here with 256 grams of carbon-14. Its half-life is that many years. Now I've grabbed some of 16 with it. How much has remained after this many years? So this is my TI, this is my TF, and I've got my T1 half right here. Right? Actually, these are A's. Sorry, A, F, I, and AF. Okay, and it wants to know how much is left after that many years. So I'm going to take, and I'm going to use what I did up above there just a minute ago, where I took n is equal to the total time divided by T1 half to give me how many half-lives it's going to go over. So I'm going to take that 2,865 years, and I'm going to divide it by 5,730 years, and that will tell me how many half-lives. So somebody that has a calculator, would you do that for us? We're just going to have to squeeze it in, because 16 is right there too. 5, exactly? Awesome. Okay, so then all I have to do is use this equation right here to find out how much is left over. So I'm going to take that 256 grams divided by 2 to the 5th power. And I'm going to get how much is left over. I'm going to sort of divide this space because I've got to answer 16 on that other side. So sorry, I did a little squish there. So anybody got this one done? So 2 times 2 is 4, times 2 is 8, times 2 is 16, times 2 is 32. So I'm basically taking 256 divided by 32, right? 8. 8 grams. And I have 3 significant figures, so I probably should say 8.00 grams are left. Okay? On the other side, number 16 here says 1/8, and I'm going to change colors so we can keep track of this. So 1/8 of the original sample remains. How many half-lives have passed through? So if 1/8 that is left, then that means 2 to what power equals 8? 8 equals 2 to what power? 2 times 2 is 4, times 2 is 8, so that x is 3, so I've undergone 3 half-lives. Does that make sense? So I'm finding out how many times to get times 2 gives me 8, because 1/8 is left over. Okay? All right, so the rest is going to be yours. We are going to work on this worksheet and the nuclear decay series lab. So on the nuclear decay series lab, I've given you each, or you get to come up and pick. This is regular Skittles. This is Skittles sour and sweet. The last one has a little bit of regular Skittles in it too. And this one is the berry mix Skittles. So pick one of them up. Everybody gets one. You need a napkin. You're going to open the napkin when you get back to your desk. Count how many Skittles you've got. Put them back into the cup. Okay, and then you're going to shake them out. And the ones that are up are still radioactive. The ones that are down are now safe, and you put them away. So on your data table, if you're looking at your lab now really quickly, you've got a data table there. You're going to do this trial 3 times. So on the data table on the right side of the first page, the first trial you do, you're always going to hopefully have the same amount for number 0 trial. Hopefully you don't drop any on the floor. Okay? You're going to have the same amount all the way across. For the first trial, you're going to keep dumping it, writing down how many you have still up, and dumping those back into the cup, shaking them and pouring them out again, putting them in the cup again, shaking them out, pouring them out again. So grab yourself 2 napkins so you can put the ones that are face down on the other napkin. Okay, those are now safe to eat, but you can't eat them until after you've done the trial 3 times. Okay, once you've done the trials 3 times, gone all the way to 0, 3 times, no Skittles left up. Okay, you're going to pick some color pencils up, and you're going to pick 3 colors. Color trial 1, the top of that column, 1 color. 2, another color. 3, another color. And then on the back side, you're actually going to graph that on the same graph. Okay, so make sure that you're highest number is as big as possible and use all the way across. So if I only did 5 trials, I'm going to make that whole thing go all the way across to 5 trials. Okay, make it as big as possible. And you're going to take and put each graph on top of each other and see how they correspond to each other. Okay? Questions? Yes. Yes, I want the quizzes to come into your box. So as we get up and move around, grab your colored pencils, grab your Skittles and your 2 napkins and head back to your desk.